Last week, I was sharing on the first 5 of the 14 best science evidence-based anti-aging supplements. Today, I will be discussing the other 5. Shout out again to longlonglife.org for compiling evidences and sharing to the world the latest and greatest information in anti-aging science. Do check them out and support their work if you haven't. Hi, my name is William and welcome to my channel where I share science-based evidences on products, claims and effective methods related to health and beauty concerns so you can make an informed buying power. If you are new to my channel, I upload a new video every Friday, so please subscribe if you haven't and click on the notification bell to get notified of new videos. And for anyone who are interested, when available, links to cited articles and research papers are listed in the description below. In my last video, I've mentioned the benefits and properties of 5 supplements, namely Curcumin, epigallocatechin gallate or green tea extract, coenzyme Q10, nicotinamide adenine dinucleotide, and resveratrol. This week, I will touch on 5 more effective supplements that benefits anti-aging humans. Quercetin is a pigment or flavonoids found naturally in many foods, such as onion, capers, olive oil, and some legumes. It is an antioxidant and fairly powerful anti-inflammatory agent, originally used in the treatment of asthma and allergic reactions. Its role has proven interesting in the fight against inflammation and the prevention of cardiovascular diseases. Today, quercetin is the subject of new studies as it seems to be important in the fight against aging and its associated diseases. First tested in cell cultures then on animals, the efficacy of quercetin on humans remains a subject of discussion within the scientific community. Quercetin supplementation seems to have beneficial effects on neurodegenerative diseases. In mice, it reduces certain protein-associated effects on the brain of a model mimicking Alzheimer's disease. Quercetin increases learning performance and spatial memory to degrading function in Alzheimer's disease. Combined with physical exercise, quercetin also has a defensive role against neuron loss. This action seems linked to the upregulation of a signaling pathway involved in the antioxidant and anti-cell death activity of neuronal cells. New studies suggest that quercetin could reduce cell senescence or aging, a phenomenon affecting part of our cells, preventing them from dying but at the same time restricting their normal functions. In cultures of human aged cells, a team recently proved an inhibition of this phenomenon, leading the cells to an old phenotype thanks to quercetin. More impressively, on cells supporting the skin, a second study showed a reversal of aging, especially in the treatment of skin aging in the short term and also in the treatment of systemic cell senescence in the longer term. Glutathione plays a central role in maintaining the oxidizer or reducer balance within our cells. Due to its ability to exist in reduced or oxidized form, glutathione is a true electron trap, placing it very high on the list of antioxidants. Its metabolism is intimately linked to that of selenium because the enzyme that glutathione forms by coupling with selenium allows the detoxification of free radicals. Glutathione is involved in a wide variety of phenomena and indeed plays a multifactorial role in aging. Overall, there is a decrease of the glutathione levels during our lives, and this seems to impact the onset of diseases. This decline is particularly associated with the onset of Parkinson's disease and other neurodegenerative diseases. In Alzheimer's disease, several studies have demonstrated the central role of glutathione because of its antioxidant activity. An increase in the oxidized form of glutathione in the blood and brain was found in mouse models of Alzheimer's disease no matter the age of the mouse. These studies therefore seem to indicate reduced glutathione deficiency as a precursor to neurodegenerative diseases. In addition to the impact of its reduction on the incidence of age-related diseases, glutathione appears to play a role in apoptosis, the programmed death of our cells. A recent study also looked at the links between glutathione and apoptosis in the hearts of diabetic mice, showing that its presence reduced necrosis and increased apoptosis, a protective mechanism of the heart in these conditions. Glutathione is very promising for the extension of the lifespan. Preliminary studies in roundworms actually show an activation of sirtuin pathway, which is a family of proteins that regulate cellular health, with glutathione supplementation increasing the lifespan of these small worms by about 30%. It is possible to naturally increase glutathione by eating a lot of cabbage. Phosphatidylcholine is a major phospholipid very involved in the synthesis of membranes of our cells. It produced by the liver naturally helps with digestion, helps maintain skin elasticity, and is essential to brain function. Is it also involved in aging? The answer is yes, yes, yes. Simply because in young subjects it is present in greater quantities than in the elderly. When metabolized by our body, phosphatidylcholine can release a choline molecule that can be used by the brain to produce acetylcholine, a messenger for brain cells. Therefore, its supplementation can improve brain function. This has led to preclinical and clinical studies, particularly on Parkinson's disease, with the hope of finding a new therapeutic approaches. But the results have proved to be contradictory. However, in the treatment of bipolar disorders, phosphatidylcholine supplementation appears to be beneficial. New studies have also looked at Alzheimer's disease and show that if there is an imbalance in phosphatidylcholine metabolism, it can promote the disease. The main role of phosphatidylcholine in cardiovascular diseases appears to be the prevention of hypercholesterolemia. Indeed, in mild cases, phosphatidylcholine supplementation allows a decrease in circulating cholesterol. It is important to differentiate between lecithins, a form of phospholipid mixtures containing phosphatidylcholine, and the pure supplements. The word lecithin is, in biochemistry, synonymous with phosphatidylcholine, but many abuses the term in food supplements because by bearing the name lecithin, it can simply means contain a percentage of 25 to 95% of phosphatidylcholine content. Although lecithins have beneficial effect on digestive health, their concentration of phosphatidylcholine does not always seem sufficient to obtain an anti-aging effect. Inositol Hexaphosphate, or IP6, is a natural compound found in many plants, vegetables, and food with high fiber content such as cereals. We can also find it in our cells, where it plays a central role in inter- and intracellular signal transduction, cell proliferation, and differentiation. Its antioxidant properties have long been well known, and IP6 has recently been subjected to many studies regarding its preventive role for cancer and its part in reducing the size of tumors and existing cancerous spreads. On top of those major functions, it also helps our immune system, lowers cholesterol levels, and maintains proper kidney function. On top of reducing cell proliferation, IP6 helps lower the differentiation of malignant cells. Cancer is tied to cells that not only divide very quickly, but also lose some of the signals that guarantee the physiological future of a cell. Take colon cells for instance. These cells will become less and less differentiated throughout their divisions, and their phenotype will be further and further from their function. This is how normal cells lose their initial function and become tumor cells. IP6 supplementation has been shown to lower the occurrence of this degradation and to redirect tumor cells towards a closer phenotype to that of its origin. An immune system boost was also observed during IP6 supplementation, which, combined with its antioxidant effects, helps foster a toxic environment for cancer cells and takes part in their destruction. The combination of IP6 and its precursor, inositol, has shown promising effects in animals when fighting against cancer. A pilot study in late-stage colon cancer showed efficient reduction of side effects due to chemotherapy, along with the shrinkage of tumors and cancerous spreads. IP6 has various effects on human longevity. It helps to reduce hypercalcemia, hence lowers the rate of kidney stones formation. It also regulates the metabolism of zinc and iron. IP6 helps to treat metabolic diseases such as diabetes or hyperlipidemia, as well as cardiovascular diseases. It helps to inhibit plaque aggregation and affects vision, especially on age-related eye diseases, which saw great improvements in a pilot study. And it also probably had effect on neurodegenerative diseases. IP6 is well known for its action on the immune system. It activates natural killer cells, a subgroup of our white blood cells. This characteristic makes it a very powerful anti-cancer as well as a general booster for the immune system. As a whole, IP6 is a very promising molecule that demands more clinical tests in order to fully elucidate its role and action mechanisms in anti-aging. Polyamines are essential to many central cellular mechanisms such as cell growth and proliferation, maintenance of genetic stability, and autophagy. Initially discovered in semen, hence its name, spermidine is nowadays the subject of studies that demonstrate its interest in combating aging and increasing life expectancy in yeast, Drosophila, nematode, and human cell cultures. Spermidine is the most studied polyamine. It is synthesized from putrescine and serves as a precursor to spermine. Polyamines including spermidine are found in everyday foods. Good news, there's plenty in cheese, especially brie. It also found in citrus fruits, grapes, bananas, peas, lentils, and beans, spelt flour, and whole grain cereals. Autophagy is a programmed cell death mechanism that differs from apoptosis. Autophagy is a kind of cellular cannibalism. The cell, in response to external signals, will engage a degradation of its own organelles. Aging results from several mechanisms, including complex genetic and epigenetic processes triggered, among other things, by stressful and deleterious events for our cells, resulting in the activation of programmed death. Spermidine, whose intracellular concentration is decreased during aging, has a powerful antioxidant role to fight against these processes. In yeast, studies have shown that spermidine can trigger proteins that maintain the integrity of our DNA, thus reducing oxidative stress and necrosis and regulating autophagy. Other key roles of spermidine include regulating the inflammation, lipid metabolism, and regulation of cell proliferation. It also has a cardioprotective effect and fight against neurodegenerative diseases. And there you have it, the second part of the top 14 anti-aging supplements according to science. What do you think of these supplements? Are you taking any of these and where do you get them? Do let me know in the comments below. A huge shout out to longlonglife.org for a wonderful contribution to the anti-aging science and their dedication in sharing the latest information and research on aging to the world. Do check them out on the links below. And I'll see you next week for the third and final part with an added bonus.